Welcome, everyone, to the second session of our Clovertex webinar series. Our first webinar focused on Cryo-EM processing in the industry. And one of the recurring themes during the Q&A was the role of tomography in industrial settings. And it was really interesting. Many of our panelists suggested tomography is not yet widely adopted or impactful in pharma and biotech. But we got such a sheer volume of questions on this topic. It showed there's real curiosity and momentum building in the community. And given this interest, we thought it deserved a deeper dive. So on that note, let's start by getting a really clear picture of where tomography stands in industry today. I'm really delighted to welcome our two panelists today, who are both uniquely positioned to speak to the current and future role of tomo in the industry. Dr. Maryam Khoshouei from Novartis and Dr. Dimitry Tegunov from Genentech. Maryam, why don't we start with you? You've done a lot of tomo work in academia. You now work in a major pharma company. Can you introduce yourself to the audience and share your perspective on what you heard in the last webinar and how you see tomo being perceived or used currently in pharma? What's the pulse?
Thanks, Maryam. Let me first say hi to all the attendees and thank you for inviting me to speak on behalf of Novartis. And first of all, I would like to introduce myself and what I've done before, and then continue to what I think about tomography in industry. So first of all, I did both my bachelor and master's in applied physics and also nanotechnology. And then for my PhD studies, I did my PhD studies at Max Planck Institute for Biochemistry in Martin Street near Munich. This lab is very well known for the contribution that they have, the skill they have in tomography and single particle analysis. So what I did during my PhD was a technical development to adapt what was so-called phase contrast technique to transmission electron microscope. And for application part, I try to show that this technique can be usable for both single particle analysis and also cryo-electron tomography. And since 2018, I joined Novartis, which I was heavily involved in single particle work for drug discovery. I'm not saying cryo-electron tomography yet because this method is not mature enough, the same as single particle analysis, but it doesn't mean that it won't be mature anytime soon. So I'm quite optimistic that at this point also we can answer some of the questions that we can help our colleagues in drug discovery project by doing cryo-electron.
That's wonderful. Thank you. And at this point, I should probably introduce myself too. I'm Maryam Moore. I lead strategic marketing and business positioning here at Clovertex. Clovertex is a comprehensive life sciences, HPC, cloud, AI, and scientific data consulting partner. And we've been working closely with biopharma, IT, and R&D leaders for over 7 years now. Our team has really deep scientific expertise, for example, of course, in Cryo-EM, structural biology, multiomics, and more, and a proven track record delivering secure and compliant enterprise IT solutions that meet the needs of life scientists. I also wanted to introduce my co-host today, Mazdak Rajania from Cryo-ET. And we're expecting a lot of audience questions today, given all this interest. So Mazdak's going to be keeping a close eye on our audience Q&A and is going to bring your questions up to mine and our speakers' attention as these come in relative to the discussion. So please do use that Q&A box and start typing in your burning tomo questions throughout today's panelist discussion, starting right now. So I'd like to switch now to you, Dimitry. You're also at a large pharma and you've been a really vocal advocate for tomography. Can you also introduce yourself to the audience and then share some of your thoughts on, is it fair to say tomo still doesn't quite connect with mainstream pharma problems yet? And if so, why is that?
Thank you, Maryam, and also thank you, Clovertex, for having me here today. So my name is Dimitry Tegunov. I did my bachelor's and master's in computer science at the Technical University in Munich. Also almost finished a bachelor's in biochemistry there. Then I spent about a year in Madden Street in the same lab as Maryam, where we also overlapped at the time, to see all of their amazing tomography work at the time. And then I went to Göttingen to do my PhD with Patrick Kramer, where we focused on software development. And one of the more prominent tools that I developed there was called Warp, which made single particle analysis and also some parts of tomography more streamlined and more automated. And then later I added another piece of software to that package called EM to make subtomogram averages better and higher resolution. And then in 2021, I joined Genentech as a group leader with the explicit goal of figuring out how tomography can be made useful for pharmaceutical research. And that's what I've been doing for the past almost 4 years now, mostly focusing on the software side of things. And I have a wonderful colleague at Genentech who also focuses on the environmental side of things. And yeah, I think it won't surprise anyone if I say that tomography is not quite there yet in pharma. It's definitely arrived in academia, because in academia you can choose from a much broader pool of questions that you can answer with a certain technique. And cryo-electron tomography at this point is certainly a very picky technique, right? It won't take just any question from you. You will have to think very specifically of questions that work with Cryo-ET. And yeah, whereas in pharma, we have to work at the intersection of what is pharmaceutically relevant and what can be done with Cryo-ET. And especially because so far in pharma, techniques like X-ray crystallography and single particle analysis have been so dominant, and people have gotten used to answering questions using high resolution structures of individual proteins. There isn't really yet the right type of thinking, I think, in the industry, at least among the structural biologists there, to try to think of things where you don't need a 2 or at least 3 angstrom resolution structure, where you can maybe look at something like the cellular ultrastructure and get some ideas or maybe even answers from that. Because for most of proteins that are interesting to pharma, Cryo-ET is probably not going to deliver a high resolution for inside the cell, right? There are some very favorable cases like ribosomes, proteasomes, filaments, and a few other things where this works pretty much just as well as in single particle analysis. But for most things in there, it's not going to work as well, probably ever. So
that's very interesting, Dimitry. And maybe to that point, before we get to what works and what doesn't work in tomography, the 2 or 3 angstrom structures that we use in industry. Maybe Maryam and you can talk to how are they used and who are they useful for to begin with? The single particle structures, the 2, 3 angstrom structures that you get, and if you're getting them to begin with, right?
Yeah, resolution is very important for drug discovery because our first colleagues that we go with the structure at high resolution are the chemists. And why do we collaborate so closely with the chemists? Is we want to deliver a structure of the protein with compound, and then in order to understand what are the interactions, then or having those details, you really need to achieve high resolution, atomic resolution, to be able to communicate with the chemist to see what can be the potential interaction that can be picked up after seeing the structure, and basically to improve our compound. Because at the end, when we are thinking about what can be a drug on the market, it may take up to 15 years to have a drug on the market. And by doing this structural analysis, by, for example, single particle analysis, that helps everything to move faster. It doesn't mean that if we don't have any structures, we won't able to have any drugs on the market. In the past, there was no microscope used in industry. There were other techniques at some point available, but we had times that none of the structural biology techniques were available to solve any structure. And you see, we could deliver drugs on the market. But at the moment, what we can do is to make everything faster and move faster, to have better and optimized compound in a shorter time. And at the end, hopefully to shorten the 15 years to, I don't know how many more years we can shorten these timelines, but definitely to contribute to move everything faster. In tomography, as Dimitry mentioned, usually we are dealing with proteins which are challenging, which are small proteins. And if we consider in cellular context, the abundance is not that much that we can in short time deliver structure at atomic resolution. There are limitations with the technique, both in terms of microscope and also in terms of IT. But I would say at the moment, it's not possible to answer any of those drug discovery, for example, question to answer using cryo-electron tomography, but maybe at some point in years ahead, then we can answer those questions as well. Or what we can do with tomographies at the moment, if we look at cellular context, if we look at tissues, if we treat any of those organelles with compound and then see what are the changes, what can happen, but not achieving atomic resolution, this is a kind of question we can answer with the current status of cryo-electron tomography.
Great. Thank you, Dimitry, Maryam. And yeah, indeed, it's a really great point. You know, in biopharma, the focus has to be not tool specifically, but the result. You know, how can you identify and bring new drugs to market faster, no matter the technique that you use? And yeah, so what I'm hearing, it's like there's seem to be two broad categories of limitations in terms of holding tomo back. It's first of all, it's the workflow related. It's less mature, harder to scale, more complex, and that makes it tough to meet, you know, what the chemists certainly want around resolution, timelines, and cost. And then there's the scientific limitations that many current pharma targets are small and they're membrane bound and low abundance, and that's not an easy fit for the current capabilities, as you said. So, Maryam, from your pharma experience, what sort of key performance indicators does the industry care about, and how can Tomo align, or how does it not currently align with those expectations?
So at the moment, resolution matters a lot. If you want to deliver delivery structures for drug discovery, you need to achieve, and I'm back again what Marzak asked as well, is about the resolution. The higher, the better, but we also need to understand that for proteins that we are dealing, it's different than, for example, ribosomes. It's different than, for example, compared to apoferritin. You know, these are usually flexible proteins. These are usually small proteins. And if you could achieve below 3 angstrom, then the question that you can answer is more, you can answer the question more accurately. And this is the resolution range that you need. Also, in terms of the experiment itself, it cannot take very long to deliver one structure. You have to be fast as well. And this is also one of the bottlenecks that we won't go to the direction of cryo-electron tomography at the moment, because it's very time demanding. And we cannot spend that much of time for each structure, for each experiment to deliver to the project.
Yeah, well, that certainly makes sense. Just a reminder for our audience, go ahead, start asking your questions at any time through the audience Q&A window. You can also click on the chat bubble on the screen with the question mark in it to ask your questions. And again, NASDAQ is keeping an eye out for those. We do also planned ahead of time for dedicated audience Q&A at the end. But yeah, on the note of sort of getting the resolution, I think, Dimitry, you once calculated the average protein is only represented 0.3 times per tomogram, and that really struck with me. It really highlights the fundamental challenge of using tomo for many targets in drug discovery.
Right. So I don't want to take credit for that figure. Many people before me said that, but it is certainly making our life extremely challenging, right? Because we rely on having many copies of exactly the same particle and exactly the same conformation and composition to get to any kind of high resolution. And for most particles in there, that's just going to be extremely hard to obtain. And even if somehow we can make data collection 10 or maybe even more times faster than it is now, then we still have the problem of finding such a rare and possibly tiny thing in this soup of proteins imaged with extremely low signal to noise ratio, right? And if you take today's 3D classification or template matching techniques, the abundance really does not work in their favor, right? We all remember those examples from our medical statistics classes, where let's say you have an assay that can have cancer with, let's say, 100% rate, but it also has a false positive rate of, let's say, 0.01%. And you screen the entire population with that assay, then most of the cases that it detects are going to be false positives. And exactly the same thing is going to happen with in situ data with extremely rare particles, right? Your detection technique, whatever you're using, it has a false positive rate. And once the number of negative examples overweighs the number of positive examples by many, many orders of magnitude, there is really very little hope of being able to identify something with certainty. Now, probably over time, some more probabilistic techniques will be developed that assign certain probabilities to all of those particles. And then maybe downstream of that, people will figure out how to actually make use of those probabilities. But we're certainly not there yet. And also that will require still a lot more data than we can collect at the moment.
Right. Actually, and a question just came in from the audience from Sigurdur. I think it's, which kind of relates to what you're talking about. And the question is, are you hopeful about methods which help or find or add mass to target proteins in situ for Cryo-ET? Do you have any additional thoughts on that?
There certainly have been very exciting developments in the field for tagging your protein of interest with something more contrast-rich than protein. And they definitely open a lot of new doors, and they will do even more so in the future. But then you are tagging only one or maybe a few proteins in an extremely complex system with thousands of different protein species. And it will definitely help you localize them better. But yeah, if we're talking about in situ structural biology as more of a systems biology adjacent technique, then tagging just a few individual proteins in there at a time, probably not going to help you that much in that regard.
Can I also add my opinion to this question? Adding mass is very popular method for single particle analysis. But we know if we are dealing with a target which is not very well known, it's still to use AlphaFold to design which mass we can rigidly binding to our protein of interest is also challenging, because you need to make those predictions working in the lab. You need to evaluate it in terms of expression, purification, and then test it. So I'm trying to say that even for single particle, not all the time it's very straightforward method. And in cellular context, it becomes more complicated, because if you look at the protein in native state, it can be multiple conformation. The mass that you want to add maybe adapts and maybe attached to one conformation or two conformation. How do you want to deal with so many conformations that exist? So I wouldn't say no, but I would say it's very challenging.
Yeah, certainly. All right. And we have another audience question. This one's for you, Dimitry, and regarding what you're working on currently at Genentech. It's from one of our attendees in Central Europe. And they ask, could you address the yet non-unified subtomogram averaging workflow? Several research groups with their own pipelines, file conversion, and incompatibility, etc.
That's a good question. Well, our group is working on unifying a lot of that mess that exists right now in the software, on the software side of Cryo-ET. But there is a certain limit to how much you can unify and like pre-solve for your users. So Cryo-ET is usually applied, as Maryam said, to much more challenging samples, the ones that you cannot solve with single particle analysis. And that complexity, it doesn't go away just because you apply Cryo-ET to them, right? And the questions you can then ask are usually more diverse than single particle analysis. And we can certainly pre-think some of the analysis steps for the users, certainly the same ones that we do in single particle analysis. So the classification, the averaging, and so on. But there are many questions that will require very custom tailored solution for the users. And if you want to solve them in a single package or in a single piece of software, eventually you just end up developing some kind of a scripting or programming language for the user, right? So in that sense, harder problems require more diverse, more custom tailored solutions. And I don't think that part of tomography is going to go away anytime soon. Maybe eventually LLMs will become so good at writing these custom scripts, custom programs for the users that will essentially solve that problem. But as programmers or software vendors, we probably will not solve all of the questions or help our users solve all of the questions that they might want to ask about their in situ data.
Right. Yeah, for sure. All right. And we have another question coming in. I think this one will be interesting for both of you to address. Perhaps Maryam, you can start out, which is, do you think industry has an interest in using Cryo-ET for studying GPCRs for drug discovery and development?
So first of all, GPCRs, let me say a few words about GPCRs. Maybe not everyone is familiar. GPCRs is one of the membrane protein categories, which is very interesting for any pharma companies. And it is attractive target. Yes, it is. It can be interesting for to use Cryo-ET for study GPCRs, but still GPCRs is not very straightforward, even with single particle. You know, the easy ones are done, but what is left and what you can address other diseases or you can have a drug on the market, these are usually the challenging ones. And it's very challenging if you want to address it with Cryo-ET. In terms of size, it's small. In terms of conformation or flexibility, also this challenge exists. And one other thing that I didn't touch on, but it is very relevant to also cryo-electron tomography, is sample preparation, because sample preparation is also one of the bottlenecks, both for single particle analysis and cryo-electron tomography. For cryo-electron tomography, it becomes even more complicated because if you are dealing with, for example, cells which are maybe thicker than 300 nanometer, then you need additional technique to make it possible for transmission electron microscope to deliver instruction. So sample preparation is also one of the one of the aspects that everyone should think about. Even if we solve all the IT issues, if we solve all the resolution, is the sample preparation remains. This is very important. What we could show at high resolution details, any single particle project, it's because of the high quality of the protein coming out of the purification, and then vitrification is the most important after the purification, the most critical step. If you mess up with your eyes, you will never get to atomic resolution. So this is still to be investigated and to be, easier than today for cryo-electron tomography.
Exciting. Dimitry, what are your thoughts on this, and what sort of processing or biological prep improvements would help to meet these sort of constraints in pharma and become more relevant?
Right, so as Maryam said, GPCRs are extremely interesting for pharma. They are easier to drug because they are membrane proteins facing the outside of the cell, right? There are, they definitely have some computational advantages for Cryo-ET like analysis compared to other proteins in the cell. And that is, they are located in a essentially two-dimensional surface that is very easy to identify in the tomograms, the membrane. And that constrains your search space immensely. So compared to other proteins of similar size and abundance that you might find in the cell, GPCRs and other membrane proteins are much more amenable to Cryo-ET analysis and subtomogram averaging, but also they're still super small and also not that abundant. So all of the other issues still apply, even though their positional restriction to the membrane is extremely helpful.
Actually, on that note, Dimitry, maybe I can jump in there. Is it, it used to be a big problem, right, to image tomo cell surface proteins. So while we were a bit better at membrane proteins that usually apply to cellular, subcellular membranes, what about just the cell surfaces, which is what the pharma companies are after, right? They want to hit things that are on the outside.
Right. So in terms of preserving the system in its native state, of course, the outer membrane is going to be not, well, I'm assuming it's going to be not as well preserved as everything contained inside the cell, right? It is exposed to the external environment. You still have the same problem of sample thickness, Maryam already mentioned, right? Even if something is on the outside of the cell, you still have the whole thickness of the cell next to it. So that's going to put a minimum on the sample thickness in that area. And you still have to fit mill all the way to your protein of interest. And of course, you have to either get lucky or get very sophisticated with like techniques to actually stop dipping exactly the right time, reach your protein of interest.
Yeah, because you also have to probably harvest as much of the membrane as you can on the outside, which is probably also not that trivial. At the same time, I want to go back to the previous question when somebody asked about unifying the subtomal workflow, because I know you're doing some stuff in that area. So maybe you can specifically mention that. I think you're working on a specific package. I think this will get the audience excited if they hear what you're doing. And I would be also interested to hear why is Genentech supporting this? Maybe we can talk about this.
Right. So the package that you alluded to is called Relay, and that's basically a web interface that wraps around the whole work-reliant AMP pipeline and visualizes all of the results directly in the web browser as they are produced by the processing jobs in the cluster. Lets you build entire processing pipelines that can then be executed automatically. And it's just generally much more user-friendly than is currently available in the field of Cryo-ET. Of course, in a single particle analysis he solved the same problem that already been solved a long time ago by CryoSpark. They've been extremely good at it. But for Cryo-ET, this hasn't happened yet. And we hope to at least make a contribution to that with Relay. And I think it's fair to say that it's almost ready to be released. So maybe sometime in August, if we can get it debugged just enough to be comfortable to put it in hands of external users.
Sounds
Mm.
good.
And then why am I Genentech? That's a very good question. I think I am, or rather my group and the general interest in tomography at Genentech is a gamble on tomography becoming really useful for pharma in a few years, hopefully, and then us being at the forefront. And of course, there are not too many resources for gambles like that, so they're probably only possible in really large companies like Genentech or Novartis. So there are really not too many of us, but we're excited.
That certainly sounds exciting for a community if you are able to get that release out in August. So we'll look out for that. I have another question coming in, this one from Susanna, who says, is tomography interesting for the industry in the context of cellular imaging? For example, looking at major ultra-structural changes upon compound or drug usage rather than as a structural technique.
I can pick up this question because I've already mentioned a few words that, yes, you can answer those questions by treating your, for example, organelle or tissue and see which are the differences before or after treatment. This can be answered. Not necessarily you need atomic resolution, but at the moment, is that a burning question we want to answer? No, still, the projects that we are dealing, getting high resolution structure is the most important part. And yes, if this become a very critical question that you need to answer, that could be one of the technique which could help us to answer. And then this helps actually biologists and not the chemists that we are interacting at the moment, because chemists needs to understand the atomic details of each structure, but not really what is happening to the morphology, what is happening inside the cell, what are the changes that you can see. So your audience, let's say, will be different.
Right. So, Dimitry, anything to add to that?
Maryam's absolutely correct here. And maybe just to add, all of the ultra-structural questions that you can answer with Cryo-ET, you have to subtract the set of questions that you can answer with super resolution light microscopy from that set of questions, right? Because super resolution light microscopy is much, much more high throughput and much easier to do Cryo-ET. So I think in the past, people have also mentioned some possible applications for Cryo-ET in diagnostics. I really don't think that would work. Or, well, it would work, but other approaches would work better, right? Once you've already identified some distinct ultrastructural change in the cell, then you can develop assays for that. Do not utilize Cryo-ET at all. But Cryo-ET is still going to be crucial for the initial discovery of those things.
Mm-hmm. This discussion is definitely generating a lot of interest, and we've got the questions coming in thick and fast from our audience. And actually, on that note, a sort of clinical diagnosis, we have one here. What do our speakers think of the Cryo-ET and STA potential in clinical diagnosis? So, any further thoughts related to that specific question?
Well, as I said, once you have identified something and you want to really bring it into the clinic, so you want to scale it up by many, many orders of magnitude, at least as the technique stands right now, I don't really see Cryo-ET being part of that scale up. As I said, it's going to be, well, it's going to be possibly crucial for the initial discovery of what you want to develop the assay for. But once you've figured out what's actually happening, then you will employ other biochemical or imaging techniques that are much more high throughput, just much easier to put into the clinic than Cryo-ET. I don't think we will see a bunch of Titan Cross microscopes standing in the basement of hospitals anytime soon.
I also fully agree with Dimitry, and what is, for example, worth of mentioning is there are different phases in drug discovery, and where I am at the moment is R&D, so it's the research part. It's a very early phase of any project that can be a potential drug on the market. So usually cryo-electron tomography, single particle analysis, this work that we are doing is usually in the research part. If it goes to the clinical part, it's further away from what we are doing, and it's not very efficient the way that we spend time on the research part if you want to do it for a clinical part at the end.
Right. So here's another question, which actually, Stephanie, given all the challenges that we're talking about, it's a very good question, which is that we understand there are a lot of shortcomings in Cryo-ET, but what possibilities exist? What do you see in an ideal world where Cryo-ET can be adopted by the pharma? Maryam, you want to start out and your thoughts on that?
I can start with, so as I mentioned for single particle, why do we do single particle at the moment? It's because we can help our colleagues to understand in atomic details. So resolution, very important. The technique itself, it shouldn't take very long time to collect sufficient data and analyze the data. It has to be fast. So the timeline that you can have for each project to answer the question has to be as short as possible. In the past, when I was a PhD student, maybe it took me, in very easy case, up to a month to have one structure. In some difficult cases, 3 months. If you tell your colleague, I need 3 months to solve one structure, it's not going to work. So it has to be fast. So this is in terms of also throughput. You have to be fast and you have to deal with many projects within short time to help your colleagues. So throughput is very important, resolution is very important, and how easy the technique is. At the moment, we are not developing a technique in pharma companies to, in general, in our society, we are using the mature technique to answer our biological questions or our chemical questions. So this is a bit different. In academia, you have the freedom, you have the resources to have the technique more developed, but in industry, it's not really the case. What I have to mention is we are in very close collaboration with Friedrich Mission Institute, and they are doing the basic research. We are not doing any cryo-electron tomography at the moment, but they are looking into that technique, and we are always in collaboration with each other. We look how they can push the barrier, how the technique is being matured or being developed, and for sure we look outside, and we will look how the technique will be developing in the next few years. Dimitry, do you have any opinion on that?
I think that was beautiful and probably not the answer to that question. Maybe just to add, so at the moment, Cryo-ET is probably more interesting for idea or hypothesis generation before the whole pipeline thing starts. At the same time, if you've already figured out a system well enough to produce the data for your hypothesis generation, then maybe later in the pipeline, Cryo-ET could, in some cases, be useful for relatively quick structural solving, but it's definitely not a given with that technique. Single particle and X-ray crystallography are definitely much more reliable in that sense. And Maria mentioned, usually have very little time to solve a structure, right? Because structural biology is optional in pharma, right? You can very well develop a drug without ever solving a structure of it. Structures definitely make things easier, and they help a lot, but they are optional, right? And that stands in a significant contrast to how structures are perceived in academia, right? In academia, they are the very bottom of a lot of things, which is why it is relatively easy to publish structural papers very high in journals, which is why they often make the cover image. So structural biology just doesn't have the same kind of superstar status in pharma, I would say.
Yeah, that's literally a very good point. I noticed this as well many times. Dimitry, we're getting a lot of questions about applications. One was, for instance, around other particular applications that you can think of in cell or gene therapy, as well as somebody else asked, and I thought that was a really interesting question, because you're seeing the value in the fundamental, in the basic research, or just at the cusp of basic research becoming applied. What are good ways for academia and industry to work together? How can PIs team up with industry, and how can industry team up with PIs?
I can maybe start. As I mentioned, we have Friedrich Michelin Institute, and it's a big community, because people either for doing their PhD studies, postdoc, or to become PI, they look for any opportunities at Friedrich Michelin Institute, and they are very close in collaboration with Novartis. On the other hand, there are also some programs that Novartis offers. And in order, if you are interested, you want to get more information, it's good to have a look on internet. But there are positions that, especially postdoc positions, that Novartis offers. And the postdoc can come and spend a program at Novartis to pick up one of the project which is relevant for drug discovery, and they can explore, they can learn. This also can be a good collaboration, that you can bring some knowledge from outside, you can pick up some knowledge from inside, and then give you a perspective what you want to do. And if you like more academic environment, if you like more industrial environment, if you want to be still in between, then the research institute, I think, is a good option for you, but there are plenty of ways.
So that's certainly encouraging, I think, in terms of thinking of there are opportunities out there, and how can they leverage them. So here's another question from Misha Pogrushev, which is great to see you all. What is the most exciting potential for Cryo-ET in pharma in the next 5 years, perhaps in terms of classes of proteins? Dimitry, you want to start off on that one?
Maybe Mario could say more about
Maryam, go ahead.
the proteins, because I'm really more on the computational side of things.
Sure.
But maybe I could combine this question with the other one about the probabilistic models and artificial intelligence.
It's a good question because it's just come in on that front. So we can ask that next. So Maryam, do you want to briefly talk a little bit about in terms of the opportunity of the next 5 years in terms of boxes of proteins, and then we'll jump to the probabilistic models for Dimitry.
So at the moment, what I can say is we have to wait to see how the technique is developing within the next 5 years. I'm not usually very good in predictions, but I think 5 years to have it very mature for industry to pick it up, it might not be very realistic, but I would say more than 5 years, maybe 5 to 10 years, then I think it will be mature enough that we can answer some biological questions with Cryo-ET.
All right, so Dimitry, that question on probabilistic models, how will probabilistic models and AI help with this in situ cryo-ET? Can you give specific examples of questions or data processing steps?
Well, a very unspecific question is, how does the cell work? How does the whole human body work, right? That's what we want to eventually understand so that we can develop ways to change how it works to cure disease, right? And I think it's very important to remember that a biological cell is not a system of linear conveyor belts, which is how biologists usually like to depict and to understand that system. It is just a huge mess, right? There is no evolutionary incentive for it to be a system of linear conveyor belts. And then when we develop drugs, then we usually try to throw a wrench in one of those imagined linear conveyor belts, and quite often it fails, right? Right, so in that regard, I'm actually much more optimistic about what Cryo-ET might be able to contribute to figuring out this mess in the future compared to a single particle analysis or other imaging techniques. Maybe at some point we'll be able to just throw a ridiculous amount of Cryo-ET in situ data at some AI model, and it will build this kind of super messy, very probabilistic model internally to then be able to just generate hypotheses about how altering that system might work in specific ways, or you would be able to ask it for a solution to a specific problem. I'm not very optimistic about our human ability to understand that system and all of its complexity to be able to really make use of all of that data. So yeah, it might be an AI-like system. And of course, if that ever happens, or when that happens, then the order books at Thermo Fisher and JL and the other microscope manufacturers will explode, right? Because that will suddenly become extremely valuable data. Yeah, so maybe that's the future, but the future might just as well be throwing a similar AI model at other omics scale data, right? So we don't know if Cryo-ET is going to be the most useful data for that, or maybe genomics, proteomics, or maybe just a combination of all of them. But there is no telling which one is really the most important thing.
It's actually very cool. It's a very cool thought, Dimitri, and maybe along that, there are some sample prep related questions that could probably make it more efficient to get such data. So people were asking, are we stuck with fib milling, or do you see sectioning methods contribute to the sample prep, which would then make this vision probably even more powerful?
I can start maybe to mention that during my PhD, I touched also on this technique. And what I remember, actually, when I started fib milling, it was for material science, which is much easier and much easier to handle and much easier to, interpret the data and then make sense out of it. But when everything comes to Cryo-EM, it becomes even more complicated. I could remember still the old time when I was doing fib milling. If you are lucky, within a day or even longer, then you can have some useful samples that you can put it into in transmission electron microscope. At the moment, I don't know how much it developed, but I'm certain that the efficiency is not there, that we can count it as high throughput technique. So it's very, very important. But not every sample needs to to have this complicated procedure involved. There are some cells, there are some exactly cells that are thin enough that only having a good microscope equipped with energy filter, maybe you can answer some of the questions, but not at atomic details in cellular environment. So it's definitely something below 200, 300 nanometer that you can collect some tomograms and answer some basic questions. So sample preparation for me is really an issue.
And that maybe allows us to also pivot to some career-related questions, which we should maybe also spend some time in the last few minutes that we have. So Aria was asking, the number of people with single particle cryo-EM training seems to be saturated with industry demand. And tomography is currently geared towards academia. What advice do you have current structural biology students, postdocs, trainees to make themselves valuable to industry?
Do you want to start, Dimitry? And then I ask.
Yeah, no, I have a good answer to this question. But yeah, I can try. Yes, there are definitely very, very few positions in the industry that would be able to focus on Cryo-ET. Probably a few more or just doing Cryo-ET adjacent things or in situ adjacent things. I agree that single particle or kind of in vitro structural biology seems to be saturated at the moment. And with the future development of things like AlphaFold, there may be even less demand for that. I don't know. But to get a Cryo-ET gig in the industry, well, in my case, I just got extremely lucky, right? I got this beautiful data set from Julia Mohammed's group that had a ribosome with a small molecule drug in it. Then I managed to resolve that at 3.5 angstrom. And that got people in Genentech and other pharma companies as well quite excited about the potential of Cryo-ET for pharma. But that was pure luck, right? Like I could have solved any other structure to a similar resolution that would not have been exciting or pharma at all. Maybe on the methods development side, you could try to think more specifically about how you can make that technique more interesting for pharmaceutical research. And if you manage to do that, I mean, there's also pharmaceutical research happening in academia, right? So you don't have to necessarily go into the industry to do that.
Yeah. So,
Actually, one
ahead.
second, Dimitry, since you brought up the data set that you used for you had Julia's data set. There was an interesting question. Somebody was asking about the ethics of using public data sets.
To develop software.
To the ethics of using public data sets or developing software. And isn't that actually the opportunity that we should have more public data sets and actually drive software development for that? Maybe, and I think you had some very specific thoughts even in your M paper around that. So maybe you can speak to that.
Right, having more data, more good Cryo-ET in situ data in the public domain will be absolutely crucial in the future, both for developing more advanced software techniques, but also for biologists to be able to solve some things in there to higher resolution, right? Because a single lab may not be able to collect more than 1,000 tomograms a month, but there are many structural biology labs around the world. And if they could agree on at least a limited set of systems to collect data from and certain standards, then the Chan Zuckerberg Imaging Institute is currently developing huge database to collect all of those data sets from everyone around the world, and to also be able to source certain analysis results from other people, and to put all of that in everyone's hands. Now, about the ethics question, I really don't think it's an issue, especially legally, right? When you deposit something in an empire, you agree to a very specific license. And if you don't like that license, then don't agree to it, don't deposit data on Empire, right? But legally, using data from Empire, everyone can develop both open source and commercial software. Now, my group has been lucky that we've been able to continue all of our software development open source. But I mean, it's in the license text. Read it.
And it's great that Genentech plays ball with that, right? That you're developing open source, that you're in a company and you're developing open source. So you're using community software, but you're also giving back community software as a company.
Actually, Maryam, thinking about this idea, we need more data, and the data is too siloed. Academia and pharma vendors, everyone doing their own thing. What are your thoughts on, do we need some sort of a general forum, pre-competitive initiative where these different groups can work together more effectively on Tomo?
To my opinion, yes, forum, why not? Because also single particle, what we know at the moment is also sharing the knowledge in different forums that people could share their knowledge. And they could even if they share the data, if people look around, if people read the scientific papers, then they know that data is available. But if you, for some reason, you don't see that paper, you are not aware of the data itself, then you don't know, you cannot play with it. And I remember when I was a PhD student, one of the criteria that my supervisor was very keen on doing was, if you publish, I want you to also deposit the data. So I still get from time to time emails that people are still using some subtomogram averaging data that I deposited single particle. And I'm very happy because I see that the IT developed so much that what I could achieve years back, they could achieve with better quality with the same data. The data is there now since few years, but the computational part are so advanced now that they could deliver with the same data better result. And having a forum to share our knowledge, our experiences, and what you can share. I'm not talking about any confidential information or confidential data, no. But what you feel comfortable, what you think that can be shared with others, why not? If you write it down, then everyone knows, and if they are willing, they can look around. I want to quickly mention about the possibility of joining industry, what is needed and what is interesting, because I didn't have the time to.
I know, we're 2 more minutes, so please do.
I try to hurry up. At the moment, only solving this structure with single particle is not sufficient. You have to have a good skills in sample preparation, understanding of biochemistry, understanding of how you interpret your data, your structure, modeling, and then giving feedback to your biochemist colleague, to your chemist colleague. It's very important. Also using some AI to design your construct. This is also advantage. Having Cryo-ET experience, it doesn't mean that it looks bad if you have it because we don't use it in industry. Actually, it's a plus.
Very good and positive note to end on there. And my goodness, we're unfortunately out of time. We've had so many good questions, and we apologize we didn't manage to get to yours directly, but do reach out, directly after this if you've got a really burning question we'd like to get some more insights on. But thank you, everyone, for attending and participating so actively in today's webinar. And a big thank you, especially to our panelists, Dimitry and Maryam, for such a lively and insightful discussion about the future of tomo in industry. Thank you, NASDAQ, for co-hosting today's webinar. And just to let everybody know, we will be posting a recording of this panel discussion on the Clovertex website in the coming days, and we'll share that link when it's available. So keep an eye out in your email for that. And we look forward to hopefully seeing you all again at our next webinar. Thank you, everyone.